Okay guys, I'm going to give you the lecture on chapter 11 for the final exam, and the way it's going to look is that we're going to start off by talking about the central dogma of molecular biology. Just as a refresher, everybody should remember that DNA through the process of transcription makes messenger RNA, and then through the process of translation that becomes proteins. In a eukaryotic cell, this takes place inside the nucleus, and in a eukaryotic cell, this takes place out in the cytoplasm. In a prokaryotic cell, both of these take place out in the cytoplasm because there is no nucleus. Key thing here for transcription is that we have two different types of DNA sequences. We have what's up here called an enhancer. That enhancer enhances gene expression by recruiting activator proteins to bind to it. So remember, an enhancer DNA sequence recruits activator proteins to bind to it. Those activator proteins bring in RNA polymerase to bind to the promoter on the gene. So what happens is, instead of RNA polymerase floating around inside of the nucleus looking for that particular promoter, the activators place it directly on the promoter for that gene, which then causes you to go through transcription, and then you make the mRNA, which then becomes a protein. So enhancer with activator proteins increase gene expression. Now on the other hand, a silencer binds repressor proteins. So a silencer is a sequence of DNA, and that sequence of DNA binds proteins that are called repressors. These repressors block RNA polymerase from being able to bind to the promoter. So blocking RNA polymerase, you don't allow it to perform transcription. Without transcription, you don't do translation, you don't make the protein. So silencers and repressor proteins, silencer sequences and repressor proteins decrease gene expression. So we all remember the genetic code and how it works. You read the codons as listed here, and each one of those codons codes for a particular amino acid. So you can have multiple codons for one amino acid. In a eukaryotic cell, you have what is called monocistronic genes, which means that there is one mRNA made from one gene, which makes one protein. So I have a gene, gene A, you make one mRNA from that gene, you then make one protein. In a prokaryotic cell, you have polycistronic genes, where you have multiple genes that are made on multiple mRNA. You have multiple genes that are made into an mRNA that contains these multiple genes, which then goes on to make multiple proteins. So monocistronic, mono meaning one, means you have one mRNA from one gene, which makes one protein. Polycistronic, poly means many. You have multiple genes being made on an mRNA, which then makes multiple proteins. So polycistronic is for prokaryotic, monocistronic is for eukaryotic. Remember that for translation, the ribosome reads the mRNA 5' to 3', which is the opposite of what we've talked about for transcription and DNA replication. So the ribosome reads 5' to 3'. At the 5' end there's a start codon. At the 3' end there's a stop codon. What it's going to do is it's going to read the codon, find the complementary sequence on the tRNA. That tRNA sequence is called the anticodon. At the other end of that tRNA is the amino acid. It'll then move on to the next codon, find the correct tRNA for that, and then between the two amino acids it's going to form a peptide bond. You will have to remember how to form a peptide bond like we drew in lecture. Again, the codon on the mRNA, the anticodon on the tRNA, and at the opposite end of the tRNA is the amino acid attachment site. So prokaryotes, you can have transcription, and then simultaneously you can have translation because there is no nucleus to prevent the ribosomes from getting in. So you can have simultaneous transcription translation in a prokaryotic cell. In a eukaryotic cell, the mRNA is made inside the nucleus, which then has to be processed, and that mRNA then leaves the nucleus to go out and be bound by the ribosome. So you cannot, in a eukaryotic cell, have simultaneous transcription translation because of the nucleus blocking the ribosome from entering. So remember, you have a pre-mRNA and a mature mRNA, so it has to go through some processing. You have to put a 5' cap, a 3' poly A tail, and you have to cut out all the introns before that mRNA can leave the nucleus to then go out into the cytoplasm and be bound by the ribosome for translation. So here we have a difference between the ribosomes. Bacteria have a 70S ribosome, eukaryotes have an 80S ribosome, so it's larger and more complex. The 70S ribosome of bacteria resembles the 70S ribosome in the mitochondria, which is why we believe in mitochondria at one time where it was a bacterial cell that it got engulfed by a larger eukaryotic cell. And this was the theory of endosymbiosis.